Hello everyone. Today I am going to discuss with you one of the most important topic called retrosynthetic approach and how do you identify the retrosynthetic approach of synthesis of any lead molecule of your interest. For that, I have brought you one software which is openly available on SourceForge, that is Retroscheme. So first and foremost thing, the doubt which you raise is from where we need to download this software. Just go on to Google and just type SourceForge. Just click on SourceForge Retroscheme and you can download this software which is freely available on the website. So once you download the software, once it opens, how it comes is you have an sample how it displays here, how it is going to, and this is the display page, how you going to analyze. This is the everything I'll just show you. So this is the where you need to download the software and execute. And in order to execute that, you require smiles that either you can fetch from your any database or you can even draw off from the software itself. I'll show first how to get some structure. So I'm just using Belinostat from ChemBL database. I'll just click and I'll just open the ChemBL software. This is my ChemBL. I have this is an open source chemical library where I have fetched the molecular structure with smiles. So this is the canonical smiles. I have just copied the smiles and I have gone to the software. Either I can directly paste the smiles or I can click on open. Once I click on open, my web page will open. Here I can draw that structure. So, or I can directly draw from using some moiety and fetch and copy those structure back and paste in your software. This is the fundamental approach. Just click on run. Once you run, what happens? The background of the software runs and which will display the outcome in short time. So this is the fundamental way how you are going to analyze your what you say the data. So now what outcome you are going to get, that we will see later on, that will take some time. Meanwhile, what is retrosynthesis? So retrosynthesis, you have to have some basis what exactly retrosynthesis. In the process of retrosynthesis, you you create some synthetic equivalents or synthons. From that synthons, you progress towards a desired product from the starting with an reactants. These reactants are readily available synthons or synthetic chemicals, and with an intermediate that is synthetic equivalents, and you progress towards the product. This is the fundamental information where any chemist knows about. And how what exactly the retrosynthesis is, you just break the molecule into multiple small pathways and progress towards an final product. So this is what is the basic definition of retrosynthesis. So suppose you want an final compound like this, you go back, you add nitrogen and NO2 and you reduct it, oxidation, reduction, you add some, what do you say, acylation or any synthetic methodology you approach to get a product. This is a method, what is a synthon and synthetic equivalent. Further, not only this, the software can also fetch you the data of fragmentation pattern. What is a fragmentation pattern and how it helps you in analyzing the data? In order to show this, I'll just show you my one of my research paper that is which clearly defines how you can go with analysis of any known compounds, and how you can go with literature, and how exactly you can. This is my research paper, which clearly says what is the fragmentation and how a structure fragments and how you can fetch your data. So let us wait for some time to see the data set, and meanwhile let us even go back our product. So it's still running. Let it take some time. So this is now we got the access of the soft paper. Let us see what exactly is defined in the publication, and how the this is a mass spectra where the desired product is fragmented based upon the hard fragmentation, and how you have got the results, and these are the fragments. That means these are the desired fragments from which a final product can be synthesized, or which fragmentation leads to an synthetic equivalents or synthons or fragments or molecular ions, or what do you say, different types of fragments. These fragments may be of a carbocation, maybe a carbanion, whether it may be stable, unstable, and different approaches. For that, I suggest you to go and refer my lecture series on mass spectroscopy. You have a lectures. You just go on to my lecture series and you refer to mass spectroscopic data. So that will fetch you the basic understanding of what you say carbocation, carbanion, how exactly it is going to identify the most stable carbocation, most stable carbanion, and many more things you will able to learn from the lecture. Which will this is my lecture. I'll just give you an insight how exactly you can identify the various structural fragmentation, how you can explore the carbocations, how you can explore carbanions. You can see which is most stable carbocation. If you see the structure, you have a base peak, you have a molecular ion peak, you have a fragments. These all fragments. Using these structural fragments also, you can go back and react them to get the product, or you can identify the fragmentation pattern from the lead to the reactants. This is the basic uh, chemical reactions which you need to understand. To understand this, go back to my lecture and identify the fragmentation pattern of the data. Let us see, still the software is running in the background. So, and how this software will help you in analyzing the data or retrosynthetic approach. This software will screen the available data sets in various literature or open source. Suppose, see here, this zinc database is already searched, and it identifies a near similar molecule, and its fragmentation or the synthetic pathway which is already deposited in the literature is being fetched, and that data set will be executed into the retrosynthetic fragments, and that will be presented on the data. Even if you input some structural targets which is not deposited on the data set, it will give you the template or fragments, so that from which you can initiate a reaction to go towards the product. This is the most important, basic, fundamental in any chemical reaction. Suppose if you are starting a reaction without having an information how to progress it, what are the literature of the molecules, if you have do not have a basic understanding or basic availability of the reaction protocols, so that is a very big nightmare for a chemist. In order to overcome those, you should have a designed data set, designed reactions. You have to explore literature search. You have to explore many more things to get the data. So let us see my paper, what it says. This is the fragments of a structure which will depends upon hard fragmentation in a mass spectrometer. So these are the fragments which are yielded. Now, the much more important question is whether our retrosynthetic pathway is going to fetch you this aniline, whether it's going to fetch you some sulfonamide groups. This is a matter of interest. In some pathways, even you can find some different structures than your retrosynthetic approach. Why that is happening? Because of some kind of rearrangements which are already reported in the literature. You can even see in my mass lecture, I have explained about what type of rearrangements can be possible and how from benzene you get troponium. So there are several isomerization, racemization, and what you say, you can have your structural characteristics. So what happens? You will get a different equilibrium products or synthetic equivalents. However, the synthon approach will fetch you much more time and resources and will guide you in a proper way to identify the best possible target approach in designing a reactor. Now, my software execution is completed. Okay, now it clearly says my data set is ready. Precursors are presented. Let me view it. Upon viewing, you can see what it says. This can go start with aniline. You interact with some sulfonyl chloride and get your product. So why I have explained you my research paper, and I have, you can see this is aniline. Again, you have a reaction mixture. So this is the best possible pathways or approach. Further, again, you can use this molecule as a input, and further you can progress towards further reaction steps in identifying your library of data sets of reaction mixtures or synthetic equivalents to explore much more in your research. Thank you.